Okay, so in this video we're going to set up a PCR, and the first thing to do before we pick up any pipettes or things is to do the calculations. So first, how much liquid is in each reaction? That's going to be 50 microliters. We're going to do 6 reactions. However, we're not going to make 300 microliters of reaction mixture. We're going to make a little bit extra, enough for 7 reactions. So that's going to be 350 microliters of reaction mix, also known as master mix. Okay, so we start with our 10 times buffer. We want that to be a 1 times final concentration. Our DNTPs, we want those at 0.2 millimolar. Forward and reverse primers are both going to be 1 micromolar. Water doesn't really have a concentration. Polymerase, we want 2.5 units per 50 microliter reaction. Okay, so now we do the sums. So some of these we can just do in our head. So we did 10 times dilution of the buffer, that gives us 35 microliters. It's a 50-fold dilution of the DNTPs and the primers. So 350 divided by 50 is 7. We next figure out the polymerase, and this is 3.5 microliters. And then the last thing to do is to figure out how much water to add. So the water is just the amount that's left when you take away all the other volumes from the final volume. So 350 minus 35 minus 7 minus 7 minus 7 minus 3.5 gives us 290.5 microliters of water. Okay, so now we know what we have to measure. We can proceed to the next stage. So most of these reagents are going to come out of the freezer, so it's important to thaw them first. So what we want to do is thaw them out so they're all liquid, not ice. But then we're going to keep them cold once they've been thawed. So this blue block that you can see here is a cool block, so it's going to keep everything nice and cold, but it won't keep it frozen. At least not once we've thawed it out intentionally. So we're going to get each of our reagents and thaw them as the first thing to do. Note also the other thing we're doing here is just getting all of our things we're going to need ready in front of us so they're close to hand. So here's our 10 times buffer. So we're just rubbing it between our fingers to make sure it's thawed. So Shang here is just demonstrating it very quickly. You probably need to actually rub it in your fingers a little bit longer than this to make sure that all your reagents are thawed out. You could also put them in a water bath to thaw them. You wouldn't want to use very hot water, but just room temperature water or 37 degrees is fine in a little floaty. There's a number of ways you can thaw these out, but the important thing is we want to make sure they're all liquid before we start pipetting. And then once they're liquid, we're going to put them back in the cold to keep all the reagents nice and stable. So we've got our buffer, our DNTPs, our 2 primers. Oh, that one looks like it actually has a little bit of ice left in it maybe. So hopefully that will thaw before we actually start pipetting. The water, okay, that's the second last thing. That's milli-Q water, so very pure water. We wouldn't just use tap water for PCR. And finally our polymerase, the TAC enzyme. That actually won't freeze in the freezer, so it should be liquid right from the start. Very important, label before starting. So the first tube we're going to label is our master mix tube, so we'll just put an MM on that. Okay, we're going to assemble the master mix just at room temperature. For some kinds of PCRs, it's more important to actually assemble the master mix in the cold, so on ice or in a cold block. But depending on the PCR, you can usually get away without assembling it in the cold. Ice causes some problems because if the ice melts and it gets into the reactions, they won't be sterile anymore. So we're going to label the tubes on one side with the numbers, 1 to 6 in this case, and then we're going to put our name on the other side. Shang's just putting an S here. That's sufficient to identify those tubes. Note that we're also handling the tubes carefully. We don't want to put our fingers into the inside of those PCR strip tubes. So these start out sterile, we want to keep them sterile. Okay, time to assemble the master mix. So it is somewhat important to add the reagents in the correct order. There are a few different correct orders, but there are also some very wrong orders. The way we're going to do it here is the same order that they're written down on our instruction sheet on the left there. So we're going to start with the reaction buffer, 10 times reaction buffer, 35 microliters of that. Note also that Shang's been careful opening and closing all the little Eppendorf tubes, so in a nice aseptic manner, not putting our fingers inside the tubes, just touching the edge of the lid to open it. Note also the pipetting technique, holding the pipette nice and vertical, disposing of the tips correctly, getting them out of the tip box correctly. Checking all the volumes before pipetting, so the next one is 7 microliters, and using the right pipette for each job. So this one's a P20 for the 7 microliter pipetting reaction. Next one's DNTPs. Note also with the pipetting technique, she goes down to the first stop on the pipette to suck up the reagent, but then as she dispenses, she's going to go down to the second stop, there it is, to dispense every last drop of liquid into the tube. When you're dispensing very, very small volumes, say less than 5 microliters, it's actually worth checking the tip by eye just to make sure you really have sucked up that volume. Especially when it's like 1 and 2 microliters, it's really worth just having a look at the tip, making sure that everything's there where you think it is. Other things to note here is she's doing the pipetting. See how everything that she needs is in nice close proximity, so we don't have to reach way across our desk or get up to go to the freezer to get things. Everything we need is set up before we start. Also the work area is nice and neat. There's nothing here that we don't need, only the things that we do need. Another aspect of the setup to note here is see how the tubes are arranged in the cold block in nice neat rows, and then as she uses each tube she puts it into the row above. So this is a way to tell where we're up to with our pipetting. It's very easy to get confused in these procedures and forget which tube you're up to. That's one of the most common causes of errors here. So lining up all the reagents in a row and then moving them, say, up a row or leaving a gap between them is a really good way to keep track of where you're up to. You could also do it by ticking off the reagents on the list there on the left, but it's much easier just to use a system like Shang's using here, where we're just moving the tubes up one row to indicate that we've already pipetted that tube. So we're now switching over to the P1000 to pipette the water, so this is a much larger volume. And when we add this water to all the previous reagents, this is actually going to have the effect of mixing them. So that's the reason for adding the water as the second last reagent here. So all of those previous reagents, buffer, DNTPs and primers, will now get mixed up in the water, and we'll now have a nice 1x reaction mixture with respect to the buffer. And we can add our polymerase to that, and the polymerase enzyme will happily sit in that buffer. What would be bad here would be to add polymerase enzyme directly to water. The polymerase won't be happy in pure water. It likes to have all those pH buffers and salts and things present. So adding the polymerase now, just 2.5 microliters. So it's our last ingredient in the master mix. In any kind of molecular biology reaction, the enzyme is almost always the last thing that we add. And so we're going to give it a vortex now to mix it. You could pipette up and down also, that would be fine. But important that everything's mixed up before we go to the next stage of actually aliquoting out the master mix into our PCR strip tubes. So here we're just using this black plastic mat so we can see what's going on. You don't need to do this yourself if you're doing a PCR, it's just for the purposes of the video, just to get good contrast in the visuals. Okay, and we've got 50 microliter reactions, so we're going to pipette out 6 times 50 microliters. Now for this we can use the same pipette tip for every reaction, that's fine. Because it's the same thing we're pipetting, and we're pipetting into sterile tubes that have nothing else in them. However, when we were doing the previous steps, making the master mix, changing tips is very, very important. So note in those previous steps that we were changing tips every single time to make sure our reagents don't get contaminated with things from the master mix that have already been added. So it might be a bit hard to see the details of the strip tube from this angle, but we're proceeding along that tube one at a time, adding 50 microliters to each tube. So I've zoomed in a little here. Still might be a bit hard to see though, but hopefully you can see the tip is moving along from left to right. And we're going to fill up 6 of those tubes. And because we've made a little bit more master mix than we need to, there might be a little bit left over. That's perfectly fine, as long as we have enough to get all of our reactions. Okay, so the last step in setting up the PCR is to add our template DNA. This is the case in most PCRs where the variable in the experiment is the template DNA. Occasionally the variable might be something different, like we might be, say, testing different primers against the same template, or like in our experiment where we make our own polymerase, the variable is actually the polymerase, but that's actually pretty unusual for PCR. The setup we're showing here is much more standard, where we have a master mix with all of the ingredients except DNA, and then the different ingredient that gets added to each tube is the DNA itself. So because each of our DNA samples here is different, again, note we're changing tips every time. So here's the second DNA sample, and we're just pipetting 1 microliter into the tubes. It is possible to use the P20 and the yellow tip for this. Perhaps not ideal, but it's certainly okay. Again, checking by eye to see if there's really something in the tip is often worthwhile here too. Probably a P2 or a P10 and a white tip would probably be best practice for this pipetting step. So third DNA sample. Again, notice how carefully she's opening these tubes, just touching the outside of the tube with her thumbnail and then closing the tube again. Notice also the technique for getting the tips out of the box. So holding the base of the box down with the end of the pipette, using the other hand to open the lid, getting the tip out, moving the pipette away, then closing the lid again. So the tips stay nice and sterile between each of these steps that we're doing. And note with these DNA samples the method she's using to keep track of where she's up to. In this case we're leaving a gap between each tube in that bottom row. So that's our last sample. DNA sample number 5, this goes into the 5th tube. And you may remember we have 6 reactions in total, so our 6th one is going to be a negative control with no DNA added. So what comes next? We need to close the lids of these tubes. So again, we need to do this aseptically as possible. So we just want to touch the outsides of the lids and the outsides of the tubes, not the insides. Even though we're wearing gloves here, as is appropriate for doing an experimental procedure in the PC2 lab, these gloves are not sterile, so they still may contain DNA, nucleases and other things that could contaminate our reactions. So careful how we close the lids. Often these strip tubes, you really have to push them quite hard to get them to close. So we're going to go along at the end and really push down quite hard to make sure the lids snap shut before we put them into the thermocycler. Okay, and here we go, you can see the final reactions. And note they've all got the same volume of liquid in them, 50 microliters. There's 6 tubes of 50 microliters, they're all nicely labelled and ready to go into the thermocycler. Okay, so here's our thermocycler interface. We're going to select our method. And we'll check that the method looks correct before we put our samples in and start it running. So we've got our initial denaturation, then we have 30 cycles of denaturation, annealing and extension. So now that we know the cycle is correct, we're going to put our samples into the machine. Again, push them down nice and tight so they're sitting in the little sample holding metal wells, and then lock down the lid and we're ready to go. It's probably a good idea even to set up the thermocycler before you make the master mix, so you can just quickly come along and press run when you're ready to go.